name is Rachel Flynn. I'm an associate professor in the Department of Pharmacology, Physiology, and Biophysics at Boston University School of Medicine. And my lab is interested in defining the mechanisms that regulate the alternative lengthening of telomeres pathway in cancer. And so this ALT pathway relies on a type of homologous recombination known as break-induced replication to promote telomere elongation. One hallmark of the telomeres in ALT-positive cells is that they're actively transcribed, and they generate a long non-coding RNA referred to as TERA. TERA associates with both telomeric DNA and proteins, and often forms RNA-DNA hybrids, or R loops. And these R loops can be a source of replication stress and are known to cause DNA double-strand breaks that are repaired through homologous recombination. And so this led us to ask the question of whether TERA R loops cause the break-induced replication that drives ALT, or whether TERA expression is really a consequence of this more permissive environment at alt telomeres. And so overexpressing TERA from each individual telomere would be a technical challenge, and so we decided to use an auxin-inducible degron system to systematically destroy the exoribonuclease responsible for degrading TERA. And so the hope was that inhibiting the exoribonuclease would stabilize TERA and perhaps promote R-loop formation and maybe drive ALT within the cell. But exactly what regulated TERA metabolism within the cell had not been fully defined. And so in our most recent publication in FEBS Letters, we used an auxin-degron system and identified that the 5' to 3' exoribonuclease XRN2 is responsible for the degradation of TERA, at least in mammalian cells. And following auxin-induced degradation of XRN2 in a non-ALT cell line, we saw an increase in TERA on chromatin, but we didn't really detect any increase in R-loop formation, nor were there really any phenotypes associated with ALT activity. However, when we depleted XRN2 in ALT-positive cells, we saw a significant increase in TERA on chromatin and the formation of TERA R-loops. And so this increase in R-loops correlated with a significant increase in replication stress and a DNA damage response, and consequently an increase in several other phenotypes associated with ALT activity. And so together our findings highlight XRN2 as a key determinant of TERA metabolism and telomere stability in cancer cells that rely on the ALT pathway.